I am Dr. Laxmikant Pandey, Assistant Professor, Department of Biotechnology, St.Aloysius College, Jabalpur. Today I will explain the topic PCR. In this, first of all, I will talk about what is PCR and its thermal profile, then reagents for PCR reaction, and at last, the steps of PCR. PCR is basically a molecular biology method to make multiple copies of a specific DNA segment in vitro by using a special enzyme, Dna Polymerase. The credit for the development of this method goes to Carey Mullis. He discovered this method in 1983. Now, the thermal profile. If we look at the thermal profile of PCR, it has total 6 steps. The first step is initial denaturation. It takes place at 95 degrees Celsius for 2 minutes for one cycle. The second step is denaturation, which occurs at 94 degrees Celsius for 30 seconds. Third step is annealing. The temperature for annealing varies. It varies from 45 degrees Celsius to 65 degrees Celsius, and the time for that is fixed, that is 30 seconds. The fourth step is extension, which occurs at 72 degrees Celsius and its time varies. The time for extension depends upon the length of DNA which we are going to amplify. The fifth step is final extension. The temperature for final extension is same as the extension, that is 72 degrees Celsius, and time for final extension is 10 minutes. And the last step is hold, which occurs at 4 degrees Celsius and time for is infinite. In this step, from second to fourth, that is denaturation, annealing and extensions, occurs 30 to 40 times in a cyclic manner. Now, we discuss about the different reagents required for assembling the PCR reaction. For this, we total need 7 components. First is primer pair, then DNA template, Dna Polymerase, DNTP mix, magnesium chloride, 10x buffer, and the last is nuclease-free water. Nuclease-free water mainly used for making the required reaction volume. For a standard PCR, the optimum reaction volume is 50 microliter. PCR primers are short pieces of single-stranded DNA, usually 20 to 35 nucleotides in length. 2 primers are used in each PCR reaction, and they are designed so that they flank the target region. That means one primer is complementary to the sequence residing before the initiation base of the target DNA, known as forward primer, and the other is complementary to the sequence after the last base of the target DNA, that is known as reverse primer. Primers also provides its 3' free OH group for new base addition. Now the next is DNA template. A DNA template is a double-stranded DNA that is used by the Dna Polymerase enzyme as a basis of copying the DNA. A PCR template for replication can be of any DNA source, such as genomic DNA, complementary DNA, and plasmid DNA. The composition or complexity of the DNA contributes to optimal input amounts for PCR amplification. For example, 0.1 to 1 nanogram of plasmid DNA is sufficient, while if you are using genomic DNA, in that case you have to use 5 to 50 nanogram. Now, the Dna Polymerase. It is a thermostable DNA polymerase named after the thermophilic bacterium Thermus aquaticus, from which it was originally isolated. This enzyme has inherent property to withstand repeated exposure to high temperature of 94 degrees Celsius required during PCR. It synthesizes new DNA strain from the existing strain by adding DNTPs to the growing DNA. In a typical 50 microliter reaction, 1 to 2 units of DNA polymerase are sufficient. Now, DNTP mix. DNTP mix consists of 4 basic nucleotides, DATP, DCTP, DGTP, and DTTP, as building blocks of new DNA strands. These 4 nucleotides are typically added to the PCR reaction in equimolar amounts for the optimal base incorporation. The recommended final concentration of each DNTP is generally 0.2 millimolar. Higher concentration may be used in some cases, especially in the presence of high levels of magnesium ion, because magnesium ion binds to DNTPs and reduces their availability for incorporation. Next is magnesium ion. Magnesium ion functions as a cofactor for activity of DNA polymerase by enabling incorporation of DNTPs during polymerization. A typical final concentration for magnesium in PCR is in the range of 1 to 4 millimolar. Magnesium ion helps to coordinate interaction between the 3' OH of a primer and the phosphate group of an incoming DNTP in DNA polymerizations. In addition, magnesium ion also facilitates formation of the complex between primers and the DNA templates by stabilizing negative charge on their phosphate backbone. Now, next is PCR buffer. The main function of PCR buffer is to provide a suitable chemical environment for activity of DNA polymerase. For Tec DNA polymerase, a common component in the buffer is potassium ion, which promotes primer annealing. With time, ammonium ion replaces potassium ion in the buffer because ammonium ion has destabilizing effect, especially on weak hydrogen bonds between mismatched primer template base pairing, thereby enhancing the specificity of PCR reaction. In certain scenarios, chemical additives or co-solvents may be included in the buffer to improve amplification. Co-solvents of different concentration can be used to reduce mispriming and to enhance amplification efficiency by removing secondary structures. Hence, it is important to be aware of buffer compositions for success of PCR reaction. Now we try to understand mechanism of PCR reaction. In the first step, that is denaturation. In denaturation, the double-stranded DNA gets separated and becomes single-stranded. The separation happens by raising the temperature of the mixture to 95 degrees Celsius, causing the hydrogen bonds between the complementary DNA strands to break. Now the next step is annealing. In this, primer binds to the target DNA sequence and initiate polymerization. This can only be occurs once the temperature of solution has been lowered. The annealing temperature depends upon the melting temperature of the primer, and the melting temperature of primer, that is known as TM, depends upon the base composition of primer. Next step is extension. In this step, new strands of DNA are made using the original strands as a template. Tec DNA polymerase enzyme joins free DNA nucleotides together. The result of one cycle of PCR is two double-stranded sequences of target DNA, each containing one newly strand and one original strand. The cycle comprising 3 steps, that is denaturation, annealing and extension, is repeated 25 to 40 times. Now the final extension. This step is mainly used to fill in any protruding ends of the newly synthesized strands. The last step is hold. In this, PCR product can be kept at 4 degrees Celsius for infinite time. So, by using PCR technique, we can make billion of copies of desired DNA fragments within 2 to 3 hours. So students, this is all about the insights of PCR. In my next video, I will talk about its application part. Thank you.